The overall goal of this procedure is to study desensitization and sensitivity recovery of crayfish photoreceptors as a function of circadian time, using intracellular electrical recordings of photoreceptor cells in isolated eye stalks, employing the discontinuous single electrode switched voltage clamp configuration.
This method can help answer key questions in the chronobiology field, such as the circadian time dependency of some electrophysiological properties of photoreceptors in inactivation and recovery.
The main advantage of this technique is that it allows the measurement of photoreceptors' electrical activity in spite of the difficulty to isolate cells and without disturbing the intracellular milieu.
Demonstrating this procedure will be Drs. Barriga-Montoya and Araceli de la O-Martínez, co-workers of our laboratory.
To prepare the intracellular electrode, pull a glass capillary tube with a micropipette puller to obtain a thin tip with a small opening. Next, fill the pulled capillary glass with 2.7 molar potassium chloride solution by capillarity, and then fill half of the pipette with a fine injection needle. If necessary, tap the electrode pipette to eliminate air bubbles. After that, place the electrode on its holder. Connect the holder to the amplifier head stage with the amplifier. Following that, position the electrode holder head stage with a stable 3D micromanipulator. Lower the electrode until bath solution covers its tip. Select the bridge mode of the amplifier and measure the electrode resistance. Make sure that the resistance is about 50 megaohms. Null the offset current and compensate.